Thank you very much for your kind introduction. I didn't know what type of audience I will face today. Can you hear me well? Yeah. So I will do kind of a presentation of one of the areas we are doing in the lab that is kind of related with the recent grant that was just mentioned. So in the lab, we work with viruses. You can see here this typical imaging for the viruses we work. Bacteria also have, I'm a bacteriologist, so I'm interested to understand how bacteria evolve. And importantly, viruses have an important role in moving genes, moving genetic information between bacteria. Okay? This is the typical imaging of a phage, of a virus that infects bacteria that we call phages. And importantly for this talk, phages also have parasites that we call satellites. And you can see here how this capsid is smaller. Right? This is kind of the virus of a virus. It's some elements that hijack or parasitize the viruses. And one area in my lab is understanding how these viruses and how these satellites impact bacterial evolution. Oops. Okay. It has been already mentioned that the currently antibiotic resistance, it's not yet, but we anticipate that will be a problem probably in the next 1 or 2 decades, because the number of bacteria becoming resistant is increasing in a very high rate. On the other side, probably because the bacteria will develop antibiotics very quickly, companies are not very investing too much in developing new antibiotics, so it's becoming kind of a problem that we don't do anything about AMR. Some typical, very simple medical interventions can be compromised in a near future. Bacteria evolve very quickly because they have the ability to swap DNA using many different routes. And in my lab, we think that it's important to understand how these mechanisms of horizontal gene transfer of DNA between bacteria, if we want to understand how bacteria evolve. As I said, so one of the key drivers in moving genetic information between bacterial species are the phages, the viruses that infect bacteria. In a typical virus, you see what we call the capsid, and inside the capsid, it's usually the viral DNA. And we also have another structure important here, that is the tail. And the tail is important because on the bottom of the tail, that will be the structure that will bind in the surface of the bacteria to inject the DNA. So at the end of the tail, there is a structure that will recognize the specific receptors in the surface of the bacterial cells. The phage will inject, will bind to these receptors via the tail, and then the DNA will be injected. Usually when viruses inject the DNA, what happens is the bacteria will die. And we can, theoretically, it should be a video here, but I'm not sure it's happening. Okay. This is a video, this is Staphylococcus aureus, that in a population of viruses, the viruses infect the cells and the cells will lyse. The green is when the cells lyse and the DNA is outside. This is the typical, what will happen for many viruses. But there are some viruses that we call temperate phages that they have an important life cycle. When they inject the DNA, they will start replicating, and then, as we've seen, they will produce everything that is required to create these new viral particles. They will lyse the cell, the cells will lyse, and the phages will be released. But sometimes during this process, some of the phages infect the bacterium, and instead of taking the lytic cycle, they will integrate into the bacterial chromosome. And we call these cells lysogens. Lysogens are bacteria that have integrated into the genome a phage. And these are very typical for all the pathogens. Usually pathogens, you should have several phages integrated into the genome. These phages will replicate with the bacterial chromosome, but sometimes they can be induced, for example, when the bacteria feel some kind of a stress, go into the lytic cycle. So temperate phages have what we call the lytic cycle or the lysogenic cycle. And in the lysogenic cycle, these phages are extremely important because, for example, many of them can produce toxins. When we talk about the cholera toxin is in a phage that is integrated into the genome. When we talk about the Zika toxin or many other toxins, these toxins usually are from phages integrated into the genome, meaning that when a bacterium acquires one of these phages, become more virulent. So why phages are important? As I said, many of them, when integrate, can encode toxins or other virulent genes, which is important for human disease or human animal plant disease. They are key drivers in bacterial evolution. We will see in the next slide that apart from moving their own DNA, phages can also move bacterial DNA or plasmid DNA, other DNA between bacteria. So these are constant movement of DNA between bacteria using phages as vehicles. So sometimes when we have this capsid, inside the capsid is not phage DNA, but it's bacterial DNA. And that bacteria will be delivered to a new cell, and that will be promote evolution. So phages are also important on that. Phages are also important because many key discoveries, for example, maybe you heard about the CRISPR-Cas recently, there was an anti-phage strategy originally. So restriction enzymes, many of the things have been developed using phages or came from phages. And also recently, many people are trying to use phages to treat bacterial infections because of the lack of new antibiotics. This is an example that we discovered about the impact of, for example, phages doing in bacterial evolution. Some phages, so this is the chromosome of bacterium in blue. You can see here, some phages, as I said, can integrate into the chromosome. I will not give you the details, but we discovered that all this part of the bacterial chromosome, this several hundred of kilobases, so it's a lot of genetic information, can be moved by a phage from one bacterium to another. So here you might have many different things that when arrive to a new bacteria will provide to this new bacteria with new qualities or new information that can be used, and sometimes can be the origin of the evolution of a new clone. Okay, so viruses have also an impact in moving DNA between species and evolving species. As mentioned at the beginning, we also have phage satellites. So they look like a phage, but usually they have a small capsid because they are smaller in size. So they have, this is the phage capsid. Usually phages we work are 45 kilobases, and the satellites are 15. Usually it's like 1/3 of the size. And in the typical satellite phage interaction, the phage pays for everything. Okay? All these proteins that we see here are from phage origin. So satellites hijack everything from the phage. They are like the parasites of the parasites, because phages are parasites that use the cellular machinery to produce all these things. Yeah? This is the typical image I put in the first slide. This is the satellite with the satellite DNA inside, and this is the phage with the phage DNA inside. Satellites also have virulent genes, resistant genes, can also move DNA. So whatever we have said for the phages can also apply to the satellites. So in my lab, think that to really understand how bacteria evolve, we need to understand how these phages and these satellites interact. As we also mentioned, so this is there are a lot of people trying to use phages for phage therapy as a new strategies to combat infection, and also the satellites. But there is a big problem right now for the phages. It's like any phage is not even able to infect all the what we call strains from one species. So like if we were bacteria, because we are genetically different, but we are all humans, we are what we call strains. So there is no phage that is able to kill all the strains from the same species, meaning that in terms of therapeutics, they are very narrow in the range. And remember that the structure that determines what cells can be infected by a phage or by a satellite is the tail. The tail will recognize something in the surface of the bacterium, and then will bind to and inject the DNA in that bacterium. So this is the typical satellite life cycle. So I have been working for this probably 25 years of my life to get this kind of picture. It's a bit depressing to have 25 years in one slide, but it is what it is. So we have the satellites that integrate into the genome, and then we have two scenarios. A phage can infect that cell, or the phage is already inside the cell and will be activated. It doesn't matter what is the scenario, infection or induction, the phage will start replicating. As soon as that happens, the satellite knows that this is happening. So this is a very lovely mechanism that use the satellites to know that the helper phages, it's a helper phage already replicating inside the cell. When that happens, the satellite will be also active, will also replicate, and when the phages start producing everything what is required for the capsid and the tail, the satellite will hijack part of that and will produce either the phage with the large capsids or the satellites with the small ones. The cells will lyse, and then we have what we call a lysate, and in this lysate we'll have phage particles in blue, or we'll have satellite particles in red. Until recently, what we all community assume is that what is released from a cell is a functional either phage or satellite particle, meaning that any of these particles will have a capsid and will have a tail. So these particles will go to a new cell, bind to the surface through the tail, inject the DNA, and in the case of the satellite, they will integrate into the genome. I want to highlight here that until basically this year, the dogma was whatever is released from a cell, it's functional. So the viruses are completely functional, or the satellites are completely functional, meaning that they have the capsid with the satellite of phage DNA and a tail attached to the capsid. I don't know what's happening here. Something strange with the presentation. Okay, as mentioned, for many years, this interaction has been known, and the dogma again is that the satellite used to produce these small capsids and to produce the tail, the proteins that are encoded by the phage. So without the phage, the satellite cannot produce these kind of structures. Everything is paid by the phage. A few years ago, we started working in a family of satellites that we call phage-inducible chromosomal islands, or PICIs. And this is the typical PICI. They are very small in size, as we said before. And you can see in green here a couple of genes. These are genes. Every triangle or every arrow represents a different gene with different functions. The function for these two genes in green is to hijack things from the phage. This is the two proteins that are essential to hijack a lot of things from the phage, so the phage can pay everything. But we realized that it was a family represented by this one, EDL 933, that had a large number of genes here. Okay? This is called the capsid, the protease, the portal, different names. And it's important because these genes, when they appear on a phage, they are the genes that are responsible to produce the big capsid and to put the phage DNA inside the capsid. So the homologue of these genes are the ones that, when they are in the phage, are responsible to create the capsid and to package the DNA inside. But in the PICIs, we didn't know what was the role for these genes. We were a bit surprised because the initial idea was that maybe in this kind of satellite, in this new family of satellites, these genes are responsible to produce the small capsids. But if that was the case, why a satellite want to carry all these genes with just carrying a couple of genes can hijack everything from the phage? So it was kind of we didn't know the function for the satellite, and we didn't understand why the satellite wanted to carry all these genes when we might think that it's just easier put one or two that will hijack everything. We don't need to pay anything. It's easier. So we start working on that. Also, a funny thing is that this family that we call CF PICIs was very abundant on nature. It's the satellite, in terms of the satellites, the CF PICIs are the most abundant family in nature. And another interesting thing is that the same satellites, exactly the same DNA, could be found in many different species from different genera and 7 different species. It's like if we found in a virus was satellite in humans that was also present, I don't know, in cats, horses, were exactly the same one. This is very, very strange. That was surprising for us. Yeah? So we wanted to understand how the role of these genes, and if somehow these genes were responsible that this massive presence of these satellites in nature, even in different species. So to make so many years of research in a way that everybody can understand, what we realized is that when these satellites are present in a cell, what they produce is this effect, these small capsids with the satellite DNA inside. But they are released without any tail. So there were particles that were not infective. So that was the first time that a non-infective particle was described in nature. So we thought, why is that? Well, it didn't make any sense. But then we realized that some cells, other cells, when the viruses reproduce, they produce viral particles, as we show, but some viruses also have the ability to produce an excess of tails. Okay, so we mix these two things together. That we realized is that there was a novel mechanism where some cells will release these satellites that are not infected, just the capsid with the DNA inside. Some other cells will release tails, and these capsids can recognize these tails, bind to the tail, and then form an infected particle that can inject the DNA depending on the tail. Okay? The important thing it was that some of these pickies can only bind to one type of tail. Okay? But some of the pickies can bind to different tails. And sometimes these tails have the ability to inject DNA in different species, meaning that one tail can be recognized Escherichia coli, for example, and another tail can recognize Klebsiella. If one picky can bind to two different tails that recognize different bacterial species, automatically that means that that DNA can be delivered to many different species. So that was a completely novel mechanism. Something is released, is not active outside the cells. There will be so many different cells, and depending on the tail, we will create a chimeric particle, and that chimeric particle, the tail, will determine where this DNA will go. Another important thing is that we realized that the genes that produce this small capsid were similar to the genes that produce the capsid in the phage. So theoretically, evolutionary, the phage was the origin, put the genes in the CFP genome, and these genes evolve to the point that they are incompatible. So it doesn't matter, so whatever protein is produced for the picky is to produce the small capsids and can be never used by the phage, or vice versa. So that was also a very nice thing, because that was representing how a new biological entity, the CFPs, emerged from another entity. Yeah? And we wanted to know this. We are now working to understand these two things, how this incompatibility happened and how some CFPs are very specific of one tail, but others can bind to different tails, meaning that they can spread to many different species. In parallel to this study, we were approached by Google. Google DeepMind, because they are developing a system that they call curiosities. It's an artificial intelligence system that they are developing to work as a PI, as a principal investigator. So the idea is that we interact with them, and we just want it. So they say, give us kind of a question that you are working in the lab, and we will test the system to see what the system suggests that you should do. It's like kind of working as a principal investigator. Okay, so we approach the system and ask the system. We know that we have these CFPs, that they are particular because they can produce these small capsids. And the key thing that we don't really understand is like how this DNA from these CFPs can be found sometimes in multiple bacterial species. So we just is that the question we asked the eco-scientists to provide hypotheses that could explain how it's possible that the DNA, the CFP DNA, can travel to different species. Something that when we challenged this system, we already knew the answer. Okay, so unfortunately, we make the question so many years later, because if we had made the question before, our life would have been much easier. But we made the question, the system didn't know about our answer because we didn't publish anything, we didn't publish any preprint, nothing was public. And if you want to know more about both papers have been now published online in Cell, the system provides 5 main hypotheses. Yeah? And in the way the system works is even they provide kind of a score about the probability, the security that the system has in terms of what's the more, in terms of confidence. Okay? And for that, they use an ELO system that is the system that is also used for chess. So after the system created several hypotheses, the different hypotheses compete each other and they are ranked. And surprisingly for us, the first hypothesis that the system provides was we should check the ability of these CFP to interact with different cells. That was basically what our research suggested. Okay, and we were pretty shocked because basically even provide specific ideas how to test that we know that they were right. So basically the system was able to anticipate an hypothesis that was in a pretty high level because, as I said, the paper is now published in Cell. Yeah? The system also provides other hypotheses. All of them made sense. All of them were pretty good. And in fact, we classify these hypotheses as related to the original question, this presence of the same DNA in many different species. So some of them were really related to the original question, others were not. But also, some of the hypotheses were relevant to the biology in general. Maybe it's not just specific for the CFPs, but can also apply to other satellites. And you can see here that the majority of them was yes. So the hypotheses were pretty interesting, okay? And some of them definitely open new areas of research, like the work I have presented to you, but I was also highlighting here, they said that also these CFPs could move between species using another mechanism that is conjugation that we never ever thought about it, and we are now checking in the lab. What else are we doing? As I said, this is the phage proteins that are responsible to produce the phage capsid. And you can see here that the proteins that are present in the PK are very similar. Okay, this is the identity. But as I mentioned, they don't work. So these proteins, all of them work together. These proteins work together. None of these proteins is able to work with any of these and vice versa. So this is one of a specificity, and we try to understand why. To do these things, we use cryo-EM, and this is, for example, the structure of the capsid of the phage. You can see the big capsid and the proteins interacting. This is the structure of the capsid of the PK, and we see here differences in both proteins that explain how the PK produces a small capsid, how the phage produces the large capsids, and how they cannot interact with each other. This is done in collaboration with Thiago Costa, also at Imperial College in London, and now Jonas is now postdoc in my lab. So we want to understand, we have different proteins with the capsid that need to interact to produce these infective particles. This is the capsid. There is a protein here that is called the portal, the connector, and the adapter, and the tails. We are trying to understand how all these proteins interact, both in the phage and in the CFP, how they interact with the cognate partners, but we know with the non-cognate partners. And then we try to understand how some adapters from PKs can bind to one or two different cells, because we want to develop, as I will mention later, strategies to combat infection. So we are working these things with the satellites. We think that the phages might work in a similar way, and so it's not just the satellites. We have the ability to take different tails. We also think that phages can hijack tails from other phages to create also chimeric particles that will allow them to move to different species. And the idea, as I just mentioned, is that, as I said before, for the people trying to use phage therapy, the key thing is that the tails are very narrow. So one tail will bind to one protein in the surface of one cell. If the protein is there, the tail will work. If the protein is not there, the phage cannot inject DNA in that cell. Okay, meaning that even for one species, many different strains have different receptors, meaning that at the end, no phage can infect all the strains. But we have now the ability to create a particle to which we can block different tails. So theoretically, we could make mix with a particle carrying multiple tails. So basically, all the strain from one species could be infected. And we think this can be used either to treat infections, but also can be used, for example, as a diagnostic, because all these things can be labeled, and we can use them to inject DNA in a species. If the DNA is injected, it's because that species is there, so can be used also for diagnostics. As I mentioned, the right way to do these things, you have here the cryo-EM images of that's the phage. This is the protein I mentioned before, the portal that interacts with the protein here in green that is called the connector, the adapter, and the tail. And the same for the pickies. You can see that they look pretty similar, but at the end, you cannot swap them. So this protein only works with this connector. That protein will never work with that connector. And knowing the details, the molecular details of all these interactions, we can understand how these things evolve, how these things work, and we can create chimeric particles to target whatever we want to target. So basically, is that this is the final slide summarizing whatever we are doing in this area of research. We have other areas of research that we can talk later if you want. So we try to understand the molecular basis for a new mechanism of gene transfer. With this mechanism, DNA can be moved between species because these CFPs have the ability to interact or to hijack different tails from different phages infecting different species, meaning that the DNA moves between species. We think that phages and maybe other mobile genetic elements also can use this strategy to move between species. And we are trying to use all this information to challenge systems related to artificial intelligence that we think will improve the use of funds and the speed the research in the lab. This is the last Christmas, the people in the lab. This is the agencies that pay my research, the BBSRC and MRC, and Welcome from UK and DRC from Europe. And these are the main collaborators for all the project we do in the lab. Related to this CFPs is Thiago Costa, this is an expert in cryo-EM, and with other projects about the PKs or about other projects we have in the lab is these people, John Chen from Singapore, Alberto from Spain, Eduardo from Pasteur, France, and Ross from Edinburgh. And that's it from my side. I'm happy to take now questions if you have some.
Well, thank you very much, Professor. We have been treated to a complicated story. Especially from those of us who, like me, are not biologists. But I'm sure there are.
Oh, the problem is that I didn't know what's the. Okay, so I tried to make as simple as I could. So sorry.
Yes, no. Yes, you succeeded in making it as simple as possible. But right. Now, if people would like to ask questions, or if people in the audience would like to type in their questions in the appropriate box. Where is the appropriate box? There's a chat box, isn't there? Now let's have a look. Let's have a look in the chat box first. I think we have a couple of questions already. Oops, I can't uncover it, however. Hang on. I'm having trouble.
There's one from DR Hurley.
Which go ahead and read it, Martin, if you can read it.
Yeah, two questions, please. Years ago, I heard reference to the use of phages to control sources of nosocomial infection in clinical or hospital settings. How has that progressed? And are there known parasites to the SARS-CoV-2 parasite?
Okay, so the phage therapy is still in quite early days, and we have some examples now that patients that have been treated correctly, but the number is very low. So I think it's time for the field to start providing more stronger result. Okay, this is now, there is a big problem in the phage, and it's not really a problem, but it's something that we realize. So a very hot topic now in bacterial genetics is the antiphage. So it's very known now that there are more than 200 antiphage systems that bacteria have. So different bacteria have different ones, obviously, to protect themselves against the phage. So in reality, bacteria are very well protected because evolutionarily, this is how it works, okay? And we don't really know the extent, the mechanisms of this phage, antiphage war. So in that way, we are in a very early stages, yeah? So there's a lot of investment, there's a lot of interest right now. There are some examples, but definitely this is not something that can be applied to the clinic yet. So for more precise medicine. And I don't know that there is any SARS-CoV-2 parasite, but we know that there are several. So for example, herpes virus also have satellites. So many eukaryotic viruses also might have satellites. And sometimes the outcome of the infection is depending more of the satellite than the virus. So satellites are not just present in bacteria. It's a very, I think will be, whatever is a virus will have a satellite.
Yeah, thank you.
Other questions?
We don't have any more, actually.
No more. Okay. Let me ask one. So, is there the possibility that you can sequence any of the elements that you've been discussing, e.g., particularly the phages themselves and their contents? And is that something that could be useful?
We sequence the content constantly, okay? Because, in terms, for example, to know what's inside, when we said that phages can move the phage DNA or can move other DNA from bacteria, we know that because we purify what we think it's a phage. And we sequence what's inside the phage. It's not a phage DNA, it's a chromosomal DNA, it's a plasmid DNA. So this is technology that is being used now very regularly. And we call that the viral. So now people, for example, are taking samples, we purify the viral DNA. Well, we think it's a viral DNA because it's inside the capsid, and then we know what's there. Okay, so this is a very easy technology.
Okay, so does it follow then that if you can sequence some of these elements, that you can then identify the proteins that they're encoding?
Yes.
So, I mean, is that helpful in a therapeutic sense that you can? That
was a very good question. I don't know how far that went. So, the guy that did the human sequence, or one of the main fathers of the sequencing the human, was Craig Venter. After sequencing the human genome, one idea he had was he traveled around the world just trying to find viromes and sequence everything that is inside, because he thought there will be plenty of potential enzymes or whatever can be used in therapeutics. This is an idea that has been explored a lot. I don't know to what extent that moved into the clinic, but there is a lot of genetic information that we don't really know, that it has been obtained just by sequencing what is inside.
Right, but if I've understood what Google have been up to in the last few years, I mean, the codons that you can read from the DNA sequences can guide you to the protein that would.
Correct.
And Google, of course, have been wonderful in discovering how to explain the folding of many thousands of different proteins, including, as far as I understand, most of our proteins that we're made of.
Yes.
So that is, I mean, is that one avenue that people are following that could be productive?
I think it's an avenue that people are following now in terms of developing new drugs.
Yeah.
Okay, because Google has also developing not just how the protein fold, but can also test if two proteins interact, can predict. So when we have now proteins, we can a protein that we are interested in block the activity. For example, we can try to search proteins in any database that can bind and block that protein. So I presume that this is an area that people working on these kind of interactions will develop for sure.
And I don't mean to hog the conversation. If anyone...
No, this is another question here.
We have
Can
two,
you-
yeah, from Jess Kirabiru, for example. Thank you. Very interesting talk. I'm looking forward to learning more about this area. Can you just clarify the difference, please, between helper and satellite regions within a cell? Infectious.
Okay, so it's a big question in the field how satellites evolve. Okay, for many people thought for many years that they were kind of defective phages because they look like a phage. We know that now it's not the case, and they are completely independent elements. In this case, what happened is that these new elements took a part of the phage to produce the capsid, put into the DNA, and evolve independently. So you have now something like glucophage because the genes came from a phage origin, but it's a completely different entity. We try to understand how this process evolutionary, because this is kind of the creation of a new entity from a phage. But the satellites and the phages are different. They work in a similar way because one is the satellite of the other, by the name. But how they evolve, what's the origin, is different. What happens sometimes is that the satellites can take part of the genome of a phage and evolve independently to help in their life cycle. Okay.
Yes. Yeah. And then Linda White has a question. Looking at the evolution of life on Earth, could phages help to create early cells?
This is a question if phages can exist without cells. This is because phages require cells to exist. Okay. So definitely what we think or what people assume is that the early type of life that we probably had was something related more an RNA phage than, and from that evolved. But the concept of the phage already requires a cell because this phage is a parasite. So you cannot have phages in that sense without the cell. You can have cells without phages, but not the other way around.
Yeah. Then Henry Weatherburn asks, is future work with Google DeepMind AI? And if so, what is it?
We try. Unfortunately, they didn't give us the money. Okay. So we try. So we are trying to get other people involved. So what we want is it's more in terms of developing these chimeric particles. So we want to create particles a la carte. So we want a capsid that can bind to this type of cell or to another one or several or one. So in terms of analyze all these possibilities, we thought that any system, an artificial intelligence system, might help us in designing this type of particles. So now with Google DeepMind, we try, but we are trying to get funding from other companies or other people. Okay.
Finally, Anthony Clinch asks, we have practical experience of phage therapy being used to combat highly resistant bacterial infection in my family. In desperation, we turned to the Phage Therapy Center in the Republic of Georgia, who produced custom phages that saved my wife's life. Is there cooperation with this institution?
No, from my side, we don't really work. I'm not particularly interested in this part of the treatment or phage therapy. I'm more interested in the part of the basic science and understand the mechanism of gene transfer. I'm part now. I'm sure there will be other people in UK working in this area, and I know because I'm part also that this is the idea of create a big center for phage research, led by Martha Clockie from Leicester, and that they are more interested in moving things in the direction that Anthony mentioned. Create libraries for phages that can be used, can be customized. That's what I was saying. We have very good examples so far with precise medicine, with somebody is treated individually, and we have some success there. But we need to, in terms of to succeed and to replace the antibiotics, we are still a bit far from that situation.
And that seems to be all the questions that we have this afternoon.
Can I add another? But am I right in thinking from what you said earlier that the collaboration you've already had with Google AI was actually significantly helpful to you in analyzing the approaches you might you might look at?
It wasn't, because we challenged when we already knew the answer. I think it was really good for Google that could highlight the idea that the system works very well because it was already tested in the lab. Okay.
I see.
So it did work in the other way around, and that was why we're a bit surprised that Google didn't want to continue, because we think we put the co-scientists in a very nice place. But they have other. So the phage proteins are a bit tricky sometimes because the places where they interact are very flexible. And the system they developed is the AlphaFold that is extraordinary. Doesn't work very well with these flexible regions. Okay, so I think there was we understand why it was some kind of, they were not completely enthusiastic in that part, but yeah, we think it was more beneficial for them than for us. For us, it was nice to be part of that, but we already knew it would be nice to have tested the system 5 years ago. That would have been amazing.
Yes, that would have been more helpful.
Yes.
Okay.
But highlight the power of these kind of systems in proposing hypotheses. Also, it was a lot of things related to that people need to start thinking about intellectual property. Also, you have a list of hypotheses that you need to test. Also, it was but it was quite impressive to be honest.
Yes. And there was no...
We have, yes, so it's at DR Hurley piping up with perhaps Google DeepMind should have paid you for the training experience.
Correct.
Nice idea.
Well, I'm sure they'll have found it beneficial to have your feedback on the approach that you had already analysed really among your team and decided to follow, and that it was one that their system actually did throw up as an option, even if you had already decided that was the best option. But
yeah,
worth a try perhaps to see, and maybe they'll come back. You never know. But as you also hinted, perhaps they have other fish to fry or irons in the fire, whichever metaphor you like, in terms of intellectual property. Maybe they have other angles they want to pursue for a bit first.
For us, was nice as an experience, but at the end, I think it's as I said, I really love the pure basic science of these things, and whatever distract me from that, I'm not happy. So I think it was a good experience from both sides, and I think it's
sure. I mean, your field of research sounds like a complete, several lifetimes work, given that there are presumably many pathogenic bacteria in the world, and they can keep evolving almost as quickly as we can think of ways to.
They are gambling constantly. So bacterial evolution is complete gambling. You move things, and suddenly some of this gambling produce a strain that has advantages that others don't have, and they dominate. But this gambling constantly. And the good thing is that the mechanisms that bacteria use to evolve are pretty universal. So we use the Staph aureus usually as a model, but whatever so far we have discovered in Staph aureus, you see in many other species. So that's also good in terms of the research, because many of the mechanisms we work with are universals. Many different species and bacteria follow the same patterns.
So if you can figure out ways of blocking those, perhaps, or preventing them, then that could have really quite widespread.
This is what we put on the ground. I'm not really interested in blocking. I'm more interested in understanding. But you said somebody, I'm pretty sure it can be a strategy to prevent. Okay.
Yeah.
Henry Weatherburn comments that Google DeepMind has done a training exercise and will now apply it elsewhere, which is perhaps disappointing. Comment?
I think they have their investment. So for the times we met with them, just for the co-scientists, they are doing a lot of things in several things. Just for the co-scientists, they have more than 50 people working on the system. And they contacted, in the paper that we described our interaction with Google DeepMind, there are also the 2 universities doing completely different things, drug repurposing and other things that the system show to seems to be positive as well. The difference is that for the other labs, there were the initial steps. So they have the hypothesis, they were testing the hypothesis. Okay, that in our case was the opposite way. We already knew the question, the answer, and we made the question. So it was more impactful, our strategy, because proof that the system can provide an hypothesis at the highest level is completely right.
Right. Well, if we have no more questions, I just wonder if we've been rejoined by Bill. Are you back among us, Bill, or are you still at your other meeting? We had our deputy chair standing by.
I don't think so.
If he's not around, then it falls to me to say thank you very much, Professor Penadés, for a fascinating talk that will keep us all thinking for some time. And I'd love to understand more, and to wish you good luck with your future research.
Thank you.
Earlier, but not everyone may have heard, I did actually compliment you on your FRS, which you achieved last year. Congratulations on that. And also on the recent Wellcome Trust Award that you and your team have won from the Wellcome Trust. And that's mentioned in the online Imperial newspaper. So people who study that avidly, as I do sometimes, will be very pleased to see that you've got further funding, and wish you all the very best in your future research.
Thank you very much. A pleasure, and I really hope that people can at least partially follow the talk. And thanks for your kind words, and for the invitation.